Welcome to the Bio Insights, where science comes alive.
Welcome to this explainer. We're diving straight into the bustling, microscopic factory of the human cell to decode the incredible molecular machines known as enzymes. If you've ever wondered how your body manages to digest food, copy DNA, or generate energy in the absolute blink of an eye, well, you're looking at the answer. We're going to break down exactly how these cellular engines operate, what fuels them, and how they're so tightly controlled.
Let's kick things off with a mind-blowing number to really set the stage: 100 million. You see, without enzymes, the biochemical reactions required to keep you alive would literally take years to occur. But enzymes beautifully solve the biological problem of life moving too slowly. They can accelerate chemical reactions up to 100 million times faster than uncatalyzed reactions, and honestly, sometimes even faster than that. They take processes that would naturally take a lifetime and compress them into fractions of a millisecond.
All right, let's jump into Section 1, the ultimate biological catalysts. So we need to establish exactly what these catalysts are made of, because the set of enzymes produced in a cell strictly dictates which chemical reactions are even allowed to occur. Now, the vast majority of enzymes are complex, folded proteins that increase reaction velocity without being consumed themselves. But biology always has an exception, doesn't it? A few rare RNA molecules called ribozymes can actually run the assembly line too, catalyzing things like phosphodiester bond cleavage. They're much less common, but absolutely vital to our understanding of evolutionary biology.
Moving right along to Section 2, inside the active site. If we zoom in incredibly close to a single molecular machine, we find the specific area where the actual manufacturing happens. We call this the active site. It's basically a specialized pocket or cleft formed by the intricate three-dimensional folding of the long protein chain. Inside this pocket, highly specific amino acid side chains are positioned perfectly so they can directly grab onto the target molecule, which we call the substrate, and kick off the catalysis process.
Now, for a long time, scientists thought of this interaction as a rigid lock and key, but the reality is actually way more dynamic. Through what we call the induced-fit model, the enzyme dynamically shifts its shape the moment the substrate binds. It literally hugs the substrate, undergoing a conformational change that perfectly positions all those catalytic groups to force the chemical reaction to happen.
And this interaction brilliantly illustrates how the machine continuously loops through its workflow without ever being used up. It's an elegant, four-stage puzzle that resets perfectly every single time, ready to work again.
Walking through how this builds, step by step. First, the free enzyme binds to the substrate, creating an enzyme-substrate or ES complex. Second, that induced fit triggers the catalysis, converting the substrate into the final product and creating an EP complex. Finally, the newly formed product dissociates and floats away. And the best part? The enzyme immediately resets itself to its original shape, completely unchanged, just waiting to grab the next part on the assembly line.
Which brings us to Section 3, unmatched speed and precision.
To truly grasp the sheer efficiency of this microscopic process, look at the turnover number, also known as kcat. A single enzyme molecule can churn out 10,000 finished products every single second. Just imagine a physical factory are moving 10,000 times a second. That's the kind of kinetic power driving your metabolism.
But, as we know, speed without control is just a disaster. Despite that frantic pace, enzymes are impeccably precise. They operate on 3 tiers of specificity based on exact 3D geometry. Absolute specificity means the enzyme interacts with only one single substrate, no exceptions. Group specificity allows it to work on a family of similar molecules. And stereochemical specificity is so precise that the enzyme can actually distinguish between left and right-handed versions of the exact same molecule, only binding to one specific stereoisomer.
Let's look at Section 4 now, the role of vitamins.
So, many of these highly specific machines require specialized tools to function, which brings us to the crucial role of your diet. Let's get some terminology straight first. An apoenzyme is like a heavy piece of factory machinery sitting on the floor.
It has all the gears, but it's completely inactive because it's missing its power cord or a critical drill bit. It's just the protein part waiting for its non-protein partner. Once you plug that missing non-protein component in, the entire structure transforms into what we call a holoenzyme. This is the active, fully functional unit, totally ready to start churning out products. So what exactly are these missing pieces?
While sometimes that missing piece is inorganic, we call these cofactors. These are often metal ions, utilizing elements right from the earth like zinc or iron to supercharge the active site. They help draw away electrons or stabilize shapes during those rapid 10,000 per second chemical reactions. Other times, the enzyme requires a small organic molecule called a coenzyme, and this is exactly why your diet matters. These crucial coenzymes are frequently derived directly from the vitamins you eat. For example, the niacin and riboflavin from your diet are converted directly into the organic coenzymes your machinery needs to process energy.
It's really interesting to compare how these organic tools actually attach to the enzyme. Co-substrates, like NAD+, which comes from niacin, act a lot like delivery trucks. They transiently dock at the active site, drop off or pick up electrons, and then drive away in an altered state. On the flip side, prosthetic groups, like FAD derived from riboflavin, are permanently bolted onto the machine. They return to their original form after every single reaction without ever leaving the factory floor.
All right, Section 5, controlling the chaos.
With millions of these super-fast machines running simultaneously, using up metal ions and vitamin-derived coenzymes, how does the cell not just spin out of control? How does it avoid overproducing things it doesn't need? The answer is strict cellular regulation. The cell acts as a rigorous factory foreman. For activation, when it needs more of a product, it can deploy allosteric activators to turn enzymes on, use covalent modifications like phosphorylation as a biological light switch, or just synthesize more enzymes outright. But when supplies are sufficient, it slams the brakes through precise inhibition. It can use competitive inhibitors that physically block the active site, or even feedback inhibition, where the final product of the assembly line literally circles back and turns off the very first machine. Pretty brilliant, right? Finally, let's zoom all the way out for Section 6, the cellular factory floor.
Taking a look at how this entire cellular factory floor is organized, we see that rather than just floating randomly in a messy soup and bumping into things, enzymes are neatly compartmentalized. They are localized into highly specific organelles, like your mitochondria or lysosomes. This creates distinct, isolated rooms for different manufacturing processes, which ensures that a reaction building up a molecule doesn't accidentally happen right next to a reaction trying to tear it down. And this compartmentalization isn't just about traffic control. It provides the optimal physical environment. By isolating substrates, the cell can maintain the perfect, specific pH and ion concentration required for different holoenzymes to thrive. This strict organization links thousands of individual enzymes into highly efficient, purposeful metabolic pathways, seamlessly passing products from one machine to the next in a perfect sequence.
We've seen how these molecular machines use extreme precision, dynamic active sites, vitamin power-ups, and strict regulation to keep you functioning at 10,000 reactions a second. To wrap up this explainer, I'll leave you with a thought. Considering the dizzying speed and precision required for life, what happens when just a single gear in this magnificent factory stops turning? Well, often, that's the foundation of human metabolic disease. It's a stark reminder of the beautiful, delicate balance of biochemistry keeping us alive every single second. Thank you for joining me on this deep dive into the microscopic world.